So, hi everyone. Welcome to this World Wide Web on industry, academia interactions, industry, academia collaborations, and different models that that one can choose or one can choose from in terms of getting involved with academia and industry. We thought this was a very timely topic, an interesting topic at the current time, especially since our research in genetics now really starts to inform biology. We're learning about the mechanisms that underlie the psychiatric disorders, and we hopefully are starting to see how new drug or non-drug interventions could be developed based on the new knowledge that we're getting. We may also consider that the information we get from genetics may be informative for stratification of patients. At the same time, this is also interesting because we know that there is a great complexity to drug development and also a great cost to drug development. And on the other hand, we know that there is increasing focus in the research community on societal impact. And that means that academia and industry more and more realize that the best thing they can do is actually work together in a good way. Having said that, I have experienced myself that if you work together with academia or you're publishing together with people that that come from industry, people may say, well, your research is less believable because you're interacting with industry. I've had that before. So there are some issues when you consider working together with industry. And on the other hand, an additional factor is the fact that our academia pyramid in terms of talent development and career opportunities is extremely steep. So many people decide that this is not interesting for them. The level of competition that is needed to stay in academia may not be so interesting to them. And they try to find alternative ways to do research, and industry may be an alternative career for such scientists. So I'm very happy actually that we have two people here today who are experienced in both academia and industry, doing research having done research in both settings, Till Andlauer and Chia-Yen Chen. Welcome to both of you. We had a third researcher lined up for today, but unfortunately she fell ill. So I'm sure we find another time when she will be able to present her experiences. So for now, again, welcome to Till and Chia-Yen. Till, just to very shortly mention, is a principal scientist in Boehringer Ingelheim, in the Global Computational Biology and Digital Sciences department there, and is responsible for computational biology research in relation to CNS therapeutic research. And Chia-Yen Chen is the Associate Scientific Director in the Human Genetics Group at Biogen. And prior to Biogen, he was a postdoctoral fellow at Massachusetts General Hospital and the Stanley Center for Psychiatric Research at the Broad Institute. So great to have you here. And I will give the word to Till.
Okay, great. Thank you. And I decided not to present slides in the traditional way, but just tell you a bit about how we do psychiatric research at BI and how in these different steps we collaborate with academic and other partners. But first I want to make one comment on something you said, Barbara, that puzzled me a bit, that you mentioned that you had the experience that people like frowned upon you or frowned upon people when they collaborate with pharma, or if they take money from pharma, because then this somehow devaluates their research. I'm surprised by this, because what I have to say, I mean, until a few years ago, I was in academia, and not that I ever did it, but I think there was, for some people, sometimes a pressure in academia to maybe, you know, it was more important to publish well than to make every control experiment, because in the end, who cares? You know, when the paper is out, you have the paper, your career can advance in it, and often your research doesn't have consequences in a way that it's if one result is not 100% robust, I mean, other people will look at it. It doesn't matter. For us, if you do research that is not true, or I mean, if you fake anything, it's going to be very costly for our company. If we base on any idea for a drug target on a wrong hypothesis, or on a experiment that is not 100% sound, well, and then this, after a few years investing millions into a program, it turns out that there was like some wrong experimental evidence, and the entire program has to be stopped again because maybe this pillar just breaks apart. This is very, very costly, and I think the pressure is actually, I would say, is higher for us to have reliable data backing up our projects than I experienced it at academia, where of course it's ethically demanded, but otherwise it doesn't really have consequences if something cannot be replicated from your paper. That's my view on it. Anyway, we can discuss this later. I think we have, with ECTA not coming, we have some time later at the end. Let me give you a brief idea of what we do at Boehringer Ingelheim Pharma. We say BI because Boehringer Ingelheim Pharma is just too complicated to pronounce, so I'll say BI from now on. We have 6 therapeutic areas. There's CNS, and if you say CNS, we actually mean psychiatric disorders. So we currently don't do any active research in like Alzheimer's or Parkinson or multiple sclerosis or so. Psychiatric disorders, then cardiometabolic disorders. There we do things like obesity, retinopathies, and NASH. So that's a liver disease. Obesity in turn is again, of course, also related to the brain. And then we have inflammatory and respiratory disorders, cancer immunology, immune modulation, oncology. And then we have one area that is called research beyond borders, where basically everything that doesn't fit in any of the other therapeutic areas can be conducted. So we do research in various directions, but like one of the important pillars is CNS. And I think there's very few of the large pharma companies that actually do like real hands-on research in psychiatric disorders. So I'm happy that BI is one of them. In general, just to give you an idea, how do we work? We have different departments, of course. So there's this area of research, and this research has these actual therapeutic areas like CNS research. So that is one department that does the research on potential targets for future drugs. But then there's other like cross area departments that enable the drug discovery research. So there is, for example, the department in which I am in, that's Global Computational Biology and Digital Sciences. So we do computational biology, expression analysis, and so on for all of the therapeutic areas. We also have a human genetics group that supports all of the, or supports is the wrong word, that does human genetic method development and analysis for all therapeutic areas. And then we have a biobank groups that enables access to biobanks, and actually a major part of this is to work with the phenotype data to standardize the data and to harmonize the data between different cohorts and biobanks, so to make this as integrated as possible. And then we have also data and infrastructure. That means basically pipelines for verifying data, because so for us it's very important that the data is reproducible, as I pointed out in the beginning, so that all of the data that we take in is processed in a standardized way, harmonized with pipelines that can be run again and again. And there's like one large group focusing on generating these pipelines and then executing them so that we get all of the data in a database where it is findable and reusable and so on. So fair data is very important for us. We also have an NGS lab where we can run our own, where we can do our own data generation. And finally, we have digital sciences in our department. So that would be the development of any digital product. So we not only work on classical drugs, but also things like digital phenotyping, digital therapeutics, and so on. Okay, so that was now a lot about my department. Naturally, this is what I know best. But then, of course, we have other research departments like the chemistry and so on. And then we have the clinical development, which comes after a project, a target has been brought so far that we basically think everything is sound. We have a sound biological hypothesis, we have a compound and so on. And now we can start with the clinical development. This is not done by research anymore. So there's a medicine department that takes care of that, preparing the compounds for clinical trials. And then there's one more department to mention that is called TMCP, Translational Medicine and Clinical Pharmacology. And this is the department that searches for biomarkers, does patient stratification, and that also does analysis of clinical cohorts and clinical studies. So that just to give you a bit of a background and idea of how research works. And now, if you specifically look at the psychiatric disorders, it's probably clear to all of us, there has not been such a lot of innovation in the development of psychiatric treatments during the last decades. I think the most recent development was something like esketamine or so, which is not really something, a de novo molecule, but something that has just been taken up, but that something really new has been found from scratch and brought to the clinic is quite some time ago. And we try to overcome the situation. There's of course different approaches at it. We're trying to overcome it by something that we call precision psychiatry. So we consider, as probably also many of you, that these heterogeneous DSM diagnostic categories are not really helpful for drug development. Therefore, we're using RDoc-inspired transdiagnostic patient clusters that for patients that share common symptoms, for example, related to cognitive impairment or a lack of motivation or emotion problems. And we're trying to in this context to develop circuit-specific drugs. So that means we are really trying to target the biology underlying specific symptoms. So we first identify circuits that are relevant for given symptoms, and then we look at genes that could be important in these circuits, and then we see whether we can target these genes. The most advanced programs we have in this context are currently in like negative symptoms and cognitive symptoms and schizophrenia. So for example, we have a phase 3 study on a lysine transporter for treating cognitive impairment in schizophrenia. We'll see next year whether this has been successful. Well, because all of this, it's actually challenging for us to use the PGC GWASs for our purpose, because they're typically all DSM based. So of course we look at them, but it's not really according to these circuit-based, symptom-based hypothesis that we typically like to follow. What we're lacking a bit, and this is maybe also interesting for the academic cohorts, often like really large scale cohorts that not only have EHR and self-report data available, but also validated neuropsychological tests, maybe even some EEG or fMRI data or tests in combination with omics data, especially genetics, of course. So I mean, some of these studies obviously exist. I mean, I've worked with them previously in my academic life, but then they have whatever 2,000 people or so, which I mean, I know the effort required for doing an fMRI study like this, but of course, we also all know from a genetic point of view that the sample size is not sufficient. So if you have any idea of large-scale cohorts that has at least these good tests and not only self-reports, that would be potentially interesting. So, but in general, when we have these circuit-specific hypotheses, we typically validate these biological hypotheses using preclinical data, so rodent experiments, and human experiments. Of course, wherever we have human data, we use it, and we use it on top, and this is very good to have, and we would like to move as much as possible to human data, but then in the reality, of course, modifying brain circuits and studying what happens if you knock down or out a gene or so. This is, of course, typically still preclinical work, or probably will remain so. Yes, so the typical steps that we have here is first, like ideation, development of an idea for a new therapeutic concept, then forming a biological, typically circuit-based hypothesis of how targeting a certain protein, I mean, either antagonizing it or agonizing it, reducing or increasing its function, how this could modulate the brain circuit, and how this could then influence symptoms. When this is very solid and sound, and it seems like this could be true, then we start developing a compound that targets the protein in the desired way. And when it seems that this compound is really sound, that it works, that we can show, for example, in rodent experiment that it actually interferes then with this circuit and with the symptoms as far as they are accessible in a rodent experiment, and everything looks really sound, and it seems it's safe to administer this compound to various animal models and so on. Then we go into clinical development and try to bring this into the human. So now, after this, how do we collaborate in this space with academic partners? So we have large-scale collaborations. So this is, for example, biobanks. I mean, we have access to UK Biobank, but also Genomics England. Then, for example, we're a partner in FinGen, Hunt Biobank from Norway, and some others. Then we're part of some consortia and public-private partnerships. For example, the NIH AMP schizophrenia, with the aim of developing biomarkers and treatments for people at high clinical risk for schizophrenia. Then there's this EMI European initiatives, PRISM and PRISM2, where we're part of. So I think the current focus is here on the default mode network and social dysfunction. Then we have like more individual academic research collaborations. Many of them are on target identification, or even more actually on target understanding. So we already have an idea for a target for a protein, but of course we have limited capacity also for really studying the biology behind this target idea. And so in many cases, we collaborate with academic partners to advance this biological understanding and to validate basically this target idea. We also have academic collaborations with for technology and method development. This could also be, method development that can then in the future be used for such biology understanding studies. So it could be whatever, some new fluorescent proteins, or a new microscope, or whatever. I mean, any technology or method or so that makes it easier to assess. And here we have collaborations, for example, with the universities of Zurich, with Cambridge, Bristol, Beijing, Ulm, Heidelberg. In Heidelberg, for example, there's a research institute called Biomedex that is where all of the groups working there, all of the projects are fully funded by industry. So it's part of the university, but everything is funded by industry. So to really have an applied center. I already said, so the aim of these collaborations is typically to understand the biology of the targets better, but actually we also really like to form and strengthen networks with these academic partners, and also just between these collaboration partners. So we really like to bring them together. And for this, we actually also have special symposia and so on for their interaction. We also have collaborators for like software development, for example, VRVIS, that's an institute or a center in Vienna. And then we sponsor also like translational clinical studies. We have one running with King's College and another one with Yale. And then we have postdocs and PhD students that can come in from academic institutions. If, for example, we have a Yale Biomedical Data Science Fellowship where actually applications are open right now. We have with the University of Tübingen, we have an AI and Data Science Fellowship. But we also have like individual, for example, we have a postdoc with bioinformatics in Harvard. We have a PhD students from the Munich School of Data Science, the computational biology at the Helmholtz Center in Munich. So there's various opportunities actually for just doing a postdoc with us embedded in our research, or a PhD, or even an internship is possible also for master's students. Then we sometimes invite experts to talks, to workshops, to symposia, you know, something like the digital phenotyping. When we started on this, I honestly say, like, I didn't know a lot about it. So we invited academic experts to workshops to together learn what could be done, what would be the best avenue to move this forward. We have a precision psychiatry research symposium once a year that we organize together with our academic collaborators. It's unfortunately by invitation only, but like if you are a collaborator, then you're invited to the symposium. And then finally, with some selected institutions, we actually do like workshops, like, you know, we propose a scientific question, say, okay, these are the questions that interest us. And then we do something like like a world coffee or so. We invite scientists, and from this institution, we get into different groups of scientists from BI and from the institution. And then we have proposed ideas, and then each group can pick ideas and brainstorm about it. And by that, we then, you know, prioritize, we form some concrete project ideas, and then we see for the best ones that look most suitable and feasible, how can we drive them forward? How can we start a collaboration for them? We did this with Yale, I think it was last year, and we are doing this with the ICM in Paris this year. And yeah, so these are basically the collaborations. Just to give you an idea, I mean, you can also, even if you work at industry, you can also do some continue doing some general academic engagement. So, for example, I still have a lecturer position at the School of Medicine at the Technical University of Medicine. I teach genomic medicine, psychiatric genetics, genetic epidemiology there. I also supervise PhD students. So this is okay that you do this like as a hobby, and also have a collaboration with academia in this way. And also other BI colleagues do that. And now, if you concretely, this is I have one final slide actually presented that I'll prepare, that I'll like to share. And this has, at the end of my part, this has some concrete recommendations of how you can find ideas of how to collaborate with us. Now I only have to find the window to share. You know, I didn't practice this before, and of course now I cannot find the button. but I'll just try to continue talking while I'll try to find my mouse, because this is like, even if I work in the computational and digital department, what I still didn't manage to understand is how to navigate to screens. So this is really challenging for me. So I'll just finish talking, and then I'll share afterwards, because I think this is easier. So I'll share you a link with us. So if you're interested in a postdoc, I'll share you the link for this Yale Biomedical Data Science Fellowship, where the applications are currently open. Then we also have several, just in general, open positions. If you want to work with us, I'll also share the link for that. Like, we have an open postdoc position in neuropharmacology, a data steward for biobanking, a scientist, data science for precision psychiatry, but also like team lead position for data management and infrastructure. I'm going to show this to you. And then two more things. So for real collaborations, academic collaborations, there's two things I want to propose. So we have a system that is called OpenMe, Open to Experts. This is a platform, a website, which you can take a look at. And basically here, BI scientists propose, first of all, like discontinued programs. So we have compounds for proteins, compounds targeting certain proteins that we stop development on, and we hand them over to academia. So if you see anything there that you think, well, this is an interesting target, and well, they stop maybe for political reason or whatever, but I want to work on this, we are going to provide the compound to you. You can do that, or we have this open to experts. We have some scientific questions, and we post them there. And if you think, well, I think this is interesting, I'd like to work on this, I have a possible solution, you can submit a suggestion for a collaboration there. And then basically, I mean, several people do this, and we select the most interesting suggestions, and then we start a collaboration on that. And then one more thing that I hope ECTA would talk about is the Psychiatry Consortium that actually she organizes. So this organizes public-private partnerships. So basically, via this Psychiatry Consortium, you can also suggest projects, something that you think is interesting, that could be interesting for pharma. And then different pharma companies like us, like Biogen, Janssen, and so on, they are part of this consortium, and they can take up your ideas and start a collaboration as a public-private partnership. So these are the most accessible existing avenues for starting a collaboration. That's all I wanted to say, and I think I talked a bit longer than anticipated, but fortunately ECTA is not here, so it's not we're not running out of time.
Well, not fortunately that ECTA is not here, but unfortunately we're not running out of time. That's true. Thanks a lot, Till. This is very interesting, and very interesting to see how from the early beginnings, there is so much collaboration between academia and industry. The paper is open, or the discussion is now open. And as I understand it, you can either write your question in the Q&A, or you can raise your hand, and then you will be given speaker rights, and you can voice your question as well. So here is Till's slide while we talk.
Exactly, and it's always easier to, I still can't multitask. So as soon as I'm not talking anymore, I can actually click on buttons.
There is one question from Klaus Berger. Hi, Klaus.
Hi, Till.
Hi.
This is Klaus Berger from Münster University. Can you hear me?
Yes.
Okay. You mentioned your favorite large cohorts you would rather like to cooperate with instead of the small ones, even though you acknowledged the expertise and the engagement that is necessary to collect data on 2,000 instead of 200,000. So from BI's point of view, what are the most critical and the most interesting points in collaborating with these mega cohorts in terms of genetics and omics, given that most of these mega cohorts do actually have some scales or outcomes of psychiatric epidemiology and or neuropsychological testing and so on. So there are cohorts out in the world that have large sample sizes. Along with UK Biobank, there are more. And what is the primary interest for BI in collaborating with those cohorts?
Well, I mean, our primary internal interest is, of course, to, from our point of view, use this to identify either new targets for our various indications, or to understand patient subgroups better. So this is our own interest. But then, of course, we can often support each other, because then so we might be interested in certain additional data, for example, whatever proteomics, O-link data or so, that is currently not available in such a cohort. And if we think it would be valuable to have this on top, then, for example, we would organize the generation of this data. Or, for example, we have a collaboration with Gutenberg Health Study, where we then organized and financed the sequencing, the low-pass sequencing of the entire cohort. So I think in this, I mean, it's a bit of a classical model. Like we invest money on generating additional data or supporting the accessibility and the infrastructure. We give advice on what kind of phenotypes or measures would be most valuable for us. And in the end, we, together with everyone else, then get access to these generated data.
Okay, thank you. Other questions? I don't see additional questions, and my proposal would be that we give the word to Chia-Yen, and then in the end we open up for a general discussion again. Is that okay? Okay. Chia-Yen, word is for you.
All right, thank you. So I'll probably try to first share my slides, and I'll start from there.
Can you stop sharing? Yeah.
Okay. So let me, excuse me. Okay. Okay, and the slides looks fine. Okay, great. So I would like to first thank the organizers having me here to share some of my experience in working in human genetics in collaboration with academia at Biogen. So I'm Chia-Yen Chen. I'm in the human genetics group in translational sciences at Biogen. And just before I start my presentation, I would like to first briefly introduce Biogen. So Biogen is a pharmaceutical company where our focus is on CNS disorders. So we basically work on different kinds of diseases that is related to brain or nerve system. So several major areas of Biogen includes multiple sclerosis, Alzheimer's disease, and we also invest a lot into ALS, and also some early genetic and neurodevelopmental disorders. And most recently, we also start our efforts into neuropsychiatry in collaboration with Sage Therapeutics. So I think with this background and as a researcher in human genetics at Biogen, I would like to talk a little bit about how we do human genetics research at Biogen and in collaboration with academic institutes. So just a little bit background. I think this paper is particularly interesting. This is the latest paper in a line of publications of trying to provide evidence that human genetics can actually help with drug development. So this paper was published last year, which shows that if we look at the drugs that's approved by the US FDA, that's 50 of them back in 2021, we can find that about 66%, so 33 of the 50 drugs got approved in 2021, actually have human genetics evidence support. We can see that a lot of them are actually oncology drugs, but there's also drugs for other indications. So this kind of reports coming up probably back 5 or more years ago, that people keep looking into the approved drugs and find that there's actually higher chance of success for the drugs that have human genetics evidence support. And I think that's at least one of the reasons why a lot of large pharmaceutical companies, including Biogen and BI and many others that Theo mentioned, are invested into human genetics as a research tool to improve drug development. So just briefly introduce, I think the important part, probably the most important part of the human genetics group at Biogen, the type of research that we do. So we spend a lot of our effort into target identification and prioritization using human genetics research. So you can see here, this is a very simplified outline for a drug development pipeline. We go from exploratory research to preclinical studies, to clinical development, and to FDA review, and then to post-market monitoring. And the human genetics group at Biogen, we invest a lot of efforts into the very early stage, the exploratory research. So exploratory research, you can really think of it as basically the same type of research that people are doing in academic institute. We're trying to build up knowledge that with a very focused aim, that is to identify and prioritize drug targets, that is basically genes for specific indications, that's diseases. So in our efforts, here are some examples, and it is very often going into this order that we try to leverage all the human genetic evidence that's listed out here. But of course, the process is also very iterative, as you can imagine. So, but for basically all kinds of genetic studies that we can think of, especially being done with the PGC, we go from genetic associations and now also have an emphasis on diverse populations. We try to do common variant association and rare variant associations studies to identify genes associated with specific diseases or other biomarkers or endophenotype that is closely related to the disease that we're interested in. And then we try to go from genetic association to gene function. So there we leverage on the QTLs for RNA or gene expression level, protein levels, and also QTLs for other omics data type, trying to link the genetic association to the gene function. And we use various methodologies, including Mendelian randomization, fine mapping, to try to establish the causal role of specific variants and to establish the direction and magnitude of effects that in the case where we want to mitigate or interfere a certain gene function, what would be the direction and magnitude of effect if we do it that way? So this is actually a very quick and can be high throughput way to assess efficacy for a specific gene target for a disease. And finally, we also work with large biobanks and, for example, running PFAS in large biobanks to try to explore potential safety concerns and indication expansion by using these larger biobank resources. So these are just a couple of examples at high level how we use human genetics evidence for drug development, especially on the target identification and prioritization side. And of course, we use all this information, knowledge, data to also help our drug pipeline through early to late stage. But today I'll focus on the earlier part of the pipeline. So I'm actually going to briefly introduce 3 projects that we did in collaboration with academic institutes and labs, and also with other pharmaceutical companies. And I think this can serve as examples how we work together. And as you can see throughout my presentation here, that at least at Biogen, and I believe in many pharmaceutical companies now, we're very open to pre-competitive collaboration across companies and academic institutes. And the hope is really to build the future and build the foundation for potential drugs to come out. So the first one is a collaboration with Todd Lenz and Max Lam at Hillside Hospital, where we try to look into the intersection between cognitive dimensions and psychopathology. So this project is actually concluded, and a paper came out last year in Nature Communications, and there's actually more in this paper. So I would recommend you, if you're interested, in check the paper. But here I'm outlining the most relevant part for target discovery. So what we did is that we curated and QC'd 19 GWAS for cognitive traits, where Biogen contribute computation resources and some of the data into this project. And we also curated GWAS for psychiatric traits, that is mainly from PGC and from some other sources and from other consortiums for the cognitive function part, such as the SSGAC, the Social Science Genomics Association Consortium. And we curated all this GWAS result, and we ran the local genetic correlation analysis, trying to identify the overlapping part between cognitive function. So that is actually, we call it cognitive task performance, CTP, and the overlap between that and the psychiatric disorders, and the overlap between the non-cognitive factors, and the overlap between that and psychiatric disorders using GWAS data. And Max and Todd actually very cleverly designed a machine learning way to identify clusters of genomic segments that we coded meta loci across the entire genome that are highlighting the overlapping between the cognitive and non-cognitive part factors with the psychopathology. And once we identify the meta loci across the genome, we then perform the functional annotation and gene prioritization for these meta loci, trying to identify the novel mechanism and the genes lies within this meta loci. So this is a project that I think as a lot of people on this in this meeting can recognize that we start with some very academic way of looking into this data, but in the end, what we can get out of it is also interesting drug targets that we can leverage on for follow-up for new drug development. So for the results, I can highlight two panels. So here on the left, we can look at that an aggregation of the meta-loci for all the meta-loci that is shared between psychiatric disorders and cognitive test performance that is over here. We can see that when we look into the gene expression level, the gene expression level for the aggregation of the genes falls into this category that is overlapping between psychiatric disorders and the CTP. The gene expression actually goes down slightly over the lifespan. So these genes actually showed higher impact at the early stage of our life. And for one specific meta-loci, we call it as the meta-loci number 1. We identify that the genes in this meta-loci are specifically related to neurodevelopmental, neuroplasticity mechanisms. And here we identified several genes from related mechanisms that we're following up with these genes. And on the right panel, you can see here in this panel, we're showing an aggregation of genes from the meta-loci that's shared between psychiatric disorders and non-cognitive functions, where we can see that the genes actually showed higher expression level in later life. Which indicates a completely different pattern as opposed to the meta-loci that is shared between psychiatric disorders and cognitive test performance. And for one specific example here, meta-loci number 5, we identified that this meta-loci is actually annotated with GABA synaptic transmission, which we know that has a very important function into a lot of brain function and related to psychiatric disorders. So also following up on genes identified in this set of meta-loci. So this is one example where we actually collaborate with consortium leaders and leverage public resources and performed starting with a very interesting and seemingly scientific or academic exploration, but we end up with some very interesting gene targets that we are now following up on. So I think this is one example. And a second example I'm going to show here is actually based on the data that Biogen and many other pharma received from UK Biobank, exome sequencing data that is already released to public, I think more than a year ago. So for this study, we look into the exome sequencing data in UK Biobank, and we performed exome-wide burden association to identify novel genes that is associated with cognitive function in adulthood in a generally healthy population from UK Biobank. And from there, we identified several genes, and especially one of them is the KDN5B gene that you can show that it shows up as associated with multiple cognitive function related phenotypes through the rare protein truncating variant burden association across the exome. And for this study, we're trying to leverage the data that is actually supported by multiple pharma with funding support that is generated. And now it is public, so I think anyone who's interested in can also leverage the same data set. But with this pre-competitive consortium setting across multiple pharmaceutical companies, we're able to generate large data set that will allow us to perform this type of research. And for that specific example, KDN5B, we actually take a step forward and collaborate with Sanger Institute and look into specific KDM5B mouse model to show that there's actually an additive effect or a gene dosage effect for the protein truncating variant in the KDM5B gene. So we can see that there's a pretty clear trend or dosage effect between the wild type heterozygous and homozygous of the gene of the KDM5B gene that is reflected on the behavioral test in the mouse model. And we also showed a similar effect on the differential gene expression in mouse brain at embryo and adult stage. So this is sort of continuing from the pre-competitive consortium work that we have across the pharmaceutical companies, and taking that forward to further work with academic institute to follow up on specific gene that could be a drug target down the road. So, and another similar, but I think more relevant example to psychiatric disorder is that we also perform very similar analysis on depression in the UK Biobank using the exon sequencing data. And this paper is also on bioRxiv now, medRxiv now. And I would also suggest, if you're interested, take a look at this. By the way, identify one specific gene from this exon-wide analysis for depression. And we actually follow up with a lot of functional annotation and pathway or gene set analysis, trying to highlight the role of different annotations that can affect the enrichment of the rare coding variant signal in depression. And we also explored the enrichment of the so-called druggable genome or drug-gene interaction and the enrichment for rare coding variants signals annotated for those drug-related or druggable genes for depression. And in the final analysis, we also showcased that this type of rare coding variants can be used to perform patient stratification along with polygenic score. That's probably linking back to a lot of the GWAS work that's been done at PGC. But this is truly a collaborative effort that we actually leverage not only data from UK Biobank, but also from FinGen, which is also a pre-competitive consortium supported by many pharmaceutical companies, and also data shared from Million Veteran Program, which is a US government supported program. So
and then, Chen, is it then, sorry if I interrupt, but is it the cohorts that you're working with that come out of the academia industry collaboration, or is it really that you're analyzing the data together with academic colleagues? How is the collaboration aspect here?
Yeah, so I think in this specific, so I think there's two aspects. One is that because, as you can see, the data source is multiple from multiple sources, and we respect and abide to the law or privacy regulations that all the respective data stays with where they are. So for the analysis that needs to be completed by each party, they're completed at each institute. Biogen has its own cloud computing and on-premise high throughput, high performance computer cluster. So we also perform some of these analyses. Actually, for most of the exon analysis were actually done within Biogen. But for some of these other analyses, they're done at the academic by the academic collaborators here, and in respective environments. So does that answer your question?
Yeah.
Yeah. So Yeah, and here's just one last slide showing the collaborators that we have here at Biogen. And I think we just have a couple minutes left. What I want to say here is that, as you can see, Biogen is very invested into building large resources, along with other pharmaceutical companies that for all of them, eventually goes into the public domain that people can leverage on. And aside from the collaboration across the companies, we are also very keen to collaborate with academic institutes. And especially now, Biogen has a really focus on psychiatric disorders and especially on depression. We're very, very keen to find collaborators who are interested in either more focused analysis on trying to identify new gene targets for specific diseases, or just very general academic or exploratory research, as I showcased here, that all of them turns out to be publications and goes into the public domain. So hopefully that addressed some of the concerns that Barbara, you mentioned in the beginning, that the resources and the scientific work that we produce along with our academic and industry partners eventually all go into the public domain. And that's our hope, that we can help with the drug development pipeline, and we can help with the scientific community to move the science forward. So I'll stop here and see if there's any questions.
Thanks a lot. It's great to see that there are so many different ways and different models in which we can collaborate. We have a few more minutes. Are there questions from the group? And I see the group. For the moment, not. Can I ask you to perhaps reflect in a few sentences on the differences? You've all grown up in academia and you've worked in academia, and now you're working in industry as a researcher. What are the main differences, do you think, in terms of the environment, the atmosphere?
So
Chia-Yen, can I ask you? Yeah.
Yeah, I can share a little bit. I think the goal is slightly different. So I think for our purpose, the research still is very much focused on trying to help with the drug development, and that's why we have a lot of emphasis on target ID and prioritization. I think prioritization is a very interesting term, because I don't think I ever heard anyone trying to do prioritization in academic research. Prioritization is really in relation to the drug development, that if we're trying to prioritize a certain potential drug target over the other ones, it does not mean that we are not interested in the other gene targets or those gene targets are not valid. It's just that drug development is a very costly activity, and we would like to prioritize our resources into the most promising target. And we believe that human genetics has a huge impact in this respect. But,
so prioritization in particular, that's the
prioritization, I think, is I think prioritization is a concept that I learned, and later on try to communicate with my academic partners. Because honestly, depending on the company size, we can invest into a handful or a dozen different drug targets, but it's extremely hard to go beyond that. So for
focus in particular. I'm sorry, I'm cutting you short a little bit to give the opportunity. So what you're saying is in particular the focus that is there. Jill, what is it according to you? Thinking. 4:30. Yeah.
I don't know if you can hear me, but someone started drilling in the next room. So.
Oh. Yeah, we can hear you. We don't hear drilling.
Yeah, my problem is that I actually cannot understand you anymore because if someone is drilling, it's very loud.
Oh, okay. 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 Good. Well, then, given the time is up anyway, unfortunately, I want to thank the speakers again. Thanks for coming in. Thanks for giving us a little bit of an insight in what it is that is there in industry and what it is that is there in terms of the landscape of academia industry interaction. I think it's very important in going forward that we understand each other better. So thanks a lot. You've contributed to that. And thanks to those who joined the session, and hopefully many more will have a look at the session when it is on the internet. Thanks a lot and have a good day. Bye, everyone.
Thank you.